Alright guys, welcome to chapter 7. This will be our last chapter of the course. We've spent a lot of time in the hypothalamus. We spent a lot of time in the pituitary. We've learned about GnRH, LH, and FSH a little bit. Now we need to go to the ovary and the gonad and learn what happens upon stimulation by LH and FSH as far as hormone production is concerned. We know that testicles make testosterone. We know that ovaries produce estrogen and progesterone. Estrogen comes from follicles and progesterone comes from the corpus luteum, which you all know about the monthly cycles or regular estrous cycles of female mammals. And so this is going to kind of sum up our semester because I think it's important to know what exactly happens when LH and FSH bind to their receptors in the ovary or the testicle. So you're going to be overwhelmed today by biochemistry. Don't worry about it. I'm doing this to make a point and to show you kind of the intricacies involved with making a steroid. But we'll start off slow. And the reading chapter is not that long if you've looked at it. I'm going to have a bunch of additional diagrams that I'll put up on PowerPoint in these video lectures. So don't let that fool you. Not much reading, but you know, this will take us through next Wednesday. So I'm just going to start putting them up. I'm trying to get 2 lectures put up today and then 1 more tomorrow, and I'm just going to keep firing them at you. Okay, you've had a few days off, a lot of down days. We're going to end strong here, if that's okay with you guys. All right, so again, we're going to start slow. No big deal. Just starting slow. Okay. In case you don't remember from either biochem or my repro class or whatever, this is basically the nucleus of all steroids. So this is sort of the backbone of all steroids. And so there's 3 benzene rings in a little house right here. I tell my undergrads that steroids are chicken wire hormones because to me they look like chicken wire. So we got really 3 benzene rings and then a pentane ring over here, and every one of these numbers refers to a carbon atom, and these carbon atoms are numbered by biochemists for a reason, and these numbers will never change. So it's kind of overwhelming at first, but if you just think about it, let's just start here at the top, the far left. It's 1 2 3 4 5 6 7 8 9 10. Jump up here, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. There's 20 carbons in a basic backbone of a steroid hormone. And this will become a little bit clearer in just a second. So this is on your first page of your reading. I'm just going to clip these out one at a time. So just remember that. Start at the left at the top. 1 2 3 4 5 6 7 8 9 10 11 12 13 14. So you kind of go counterclockwise, counterclockwise, clockwise, counterclockwise. It'll become clear when you start looking at these steroids. Okay. All right. So here is the basic steroidogenic biosynthetic pathway. I know that all of you have seen this before. This should be nothing new to you, but you're going to have to think about it a little bit in the remaining week and a half of class. So all steroids come from the molecule cholesterol. And so there's our basic backbone. Okay, so there's 18, 19, and 20. So that would be 21, 22, 23, 24, 25, 26, and the 27th carbon. So there's 27 carbon atoms in cholesterol. The first intermediate molecule between cholesterol and the first true reproductive hormone is an intermediate called pregnenolone, which is not secreted. And basically what happened is they cut the side chain off. The enzymes removed the side chain. So we're down to 21 carbons. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, and that methyl group on top is the 21st carbon. So now you know how it kind of makes sense. Okay? Well, going from pregnenolone to what the CL produces, progesterone, all we've done is change that OH group on the 3 carbon to a double bond O keto group and move the double bond from the 5-6 position to the 4-5 position. So that's the only change between pregnenolone and progesterone. Move the double bond, get rid of that hydrogen, and convert it to a double bond oxygen. So we still have 21 carbons. That's progesterone. Okay. And then to make testosterone, we remove 2 carbons. An enzyme came in there and removed 2 carbons, number 20 and 21. Number 20 and 21 are now gone. So that leaves 19 carbons. Okay. Okay. So there's number 18 and there's number 19. That carbon right there is number 18 and that carbon number 19. So when we get to estrogen, the main steroid produced by females coming out of the follicles of the ovary, we cut off 1 more carbon. We removed number 19 here. It's gone. And we made that again a hydroxyl group. And we spread the double bonds around this benzene ring. So that's estrogen, that's testosterone, that's progesterone. All coming from cholesterol. Okay, all coming from cholesterol. Now, as a big scheme of things, you have this as well. It's just kind of looking at the forest instead of the trees. There's 2 ways that we get cholesterol to these steroidogenic cells. So this could be a cell in the testicle, this could be a cell in a follicle. Okay? So we're on the way to making androgens and estrogens or progesterone. If this is a corpus luteum cell, we'd stop here. If this is a testicle, we'd stop here. And this is an ovary, and we're looking at a follicle cell, we would go all the way to here. Okay, so here's progesterone, here's testosterone, for example, which is an androgen. There's a couple of different androgens, but testosterone is the main androgen. And obviously there's estrogen. Okay, so what does LH and FSH do? Basically, these are all enzymes. Okay, these are all enzymes. These are circles with letters through it are all enzymes. Actually, that's actually A is a receptor. A is a lipoprotein receptor. And so what LH and FSH do when they come to these steroidogenic cells, depending upon the stage of the estrous cycle or the sex of the animal, okay, but you know for sure, we know LH causes androgen production. We know LH causes progesterone production. However, LH and FSH can cause estrogen production. And so we've got A, B, C, D, E, F, G, H, I. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9 different protein, either receptor or enzymes that are regulated by the gonadotropins. Okay? So some of these hormones do not have, some of these proteins do not have to be regulated by gonadotropins. For example, an enzyme here, G, okay? The enzyme that converts pregnenolone to progesterone, this enzyme is ubiquitously produced, so it doesn't respond to LH or FSH per se. So anytime that pregnenolone is produced, boom, it gets converted to progesterone very quickly, very quickly, very quickly. Okay? But as a 10,000 foot overview, every one of these circled proteins had to be transcribed and translated from a gene. And for the most part, that is the job of the gonadotropins, is to upregulate or activate those enzymes necessary to make steroids. Okay? So that's the 30,000 foot view of a steroidogenic cell, either in the ovary of the female or the testicle of a male. Okay? All right. So first, we have to get cholesterol, because we know based on the steroidogenic biosynthetic pathway, we need cholesterol. Well, there's basically two ways a steroidogenic cell gets cholesterol. It can either bring it in from the bloodstream, and this would be in all meat-eating animals, okay, and some plant-eating animals, some omnivores as well. But you know as well as I do, omnivores like us, if you eat animal flesh, you're going to ingest cholesterol. All mammalian cell membranes contain cholesterol. So if you eat meat or eggs, you're going to consume cholesterol. And cholesterol will be brought to the steroidogenic cells in low-density lipoprotein molecules. So basically it's protein covering up cholesterol to make it water soluble so it can flow through our blood. Cholesterol is a lipid, so it's not water soluble, so it has to be covered by these lipoproteins. And so you've heard of HDLs and LDLs and VLDLs. Well, that's what we're talking about here. LDLs, pretty much the bad cholesterol, as people say. That is the apolipoprotein that carries cholesterol to cells. And so the apolipoproteins actually have receptors in the cell membrane. So like a hormone. So the apolipoprotein, like a LDL, will bind to its LDL receptor. And once it does that, it will give up cholesterol. And that's how we get cholesterol in the cells. If we don't need cholesterol right at the moment, let's say LH and FSH are not binding here, and so we don't need to move cholesterol to the mitochondria because we're not having much LH or FSH present. Okay, maybe it's between pulses or whatever. Guess what? Well, cholesterol can be stored in lipid granules as an ester. So what is a cholesterol ester? A cholesterol ester is two cholesterol molecules bound together, stuck together with an ester bond. Okay? So you could just make believe that each one of these black circles contains two cholesterol molecules. So if you can put them in here, you can get them out. But two different enzymes are needed. So you can get them in and pull them out. Put them away, pull them out. And then once you get them out, the first place cholesterol has to go to is the mitochondria. Okay? And once it gets to the mitochondria, it'll be acted upon by different enzymes, which will produce pregnenolone. Pregnenolone leaves the mitochondria, goes to the endoplasmic reticulum, where the enzymes are located to convert it to progesterone. And if this is a testicle cell, like a Leydig cell, it'll continue on to androgen. If it's a female, it'll continue on to estrogen. Okay? The other and more complex and energy-consuming method to get cholesterol, so this steroidogenic cell can produce steroids, is that these cells have the ability to make it by themselves, make cholesterol de novo or brand new. Okay. So the carbons that will make cholesterol come from acetate. Okay, so these next two slides are not meant to overwhelm you, and they're going to be hard for you to see. I just want you to appreciate the complexity of what it would take for these steroidogenic cells to make cholesterol. Oh, not yet. It'll be the next slide. So, as the first page of notes from chapter 7, from the first page of the text, obviously steroidogenic cells need cholesterol. Well, there's two sources: biosynthesis or importation from the blood, like I just showed you. So biosynthesis requires a molecule called acetyl coenzyme A, or acetyl-CoA. And the enzyme that starts putting these together is HMG-CoA reductase. Okay? And so acetyl-CoA contributes 2 carbons. Remember, we need 27 carbons to make cholesterol. So the handout, you don't have this handout because you don't need it. It's 2 pages from a biochemistry book. I just want to show you what it takes for a steroidogenic cell to make cholesterol. For most cells in the testicle and in the ovary, this will be somewhat limited because our reproductive cells really have to have a continued need to make steroids, and so cholesterol from the blood is probably the preferred method to get cholesterol to these cells. But if somebody's on a low cholesterol diet, or a mammal or a ruminant, animal is not eating a lot of plant oils that would contain cholesterol, then they would have to make it on their own. So that's the first source of cholesterol, biosynthesis by the steroidogenic cell. Number 2, okay, delivery of cholesterol esters to the cell by either LDLs or HDLs. And that's why HDLs are your good cholesterol, because they don't carry much cholesterol. They're high density, but there's not much cholesterol in them, versus a low density. It's kind of counterintuitively, the high density lipoproteins don't carry as much cholesterol, and the LDL, the low density lipoproteins, carry a lot of cholesterol. Okay, so cholesterol ester is a cholesterol with a carbon and an oxygen in between another cholesterol. Okay. Okay, so the fate of cholesterol can be 2 things. It can be esterified again, so it's seems kind of foolish, but cholesterol esters come in, they're broken apart, yielding free cholesterol. If they're not needed, they'll be esterified again and stored. Okay. Once they've been broken up and we break that bond there, when we break up the cholesterol ester and it's needed to go to the mitochondria, boom, it'll go to the mitochondria. Okay, so far we know we need cholesterol. What are the 2 sources of cholesterol for a steroidogenic cell? In the follicle of the ovary or in the testicle, the Leydig cells of the testicle. What are the 2 sources? Biosynthesis, so they can make it themselves, although it's very time consuming and energy costing, or they can simply get it out of the bloodstream as these molecules travel around in our blood called LDLs and HDLs, and they contain cholesterol from our gut, from our diet. Okay? All right, hold on to your britches. I asked Kendra the other day where her britches were, and I don't know what she thought I said, but she was unloading hay in shorts. So that's an old man's term for pants, britches. So hold on to your britches. All right, here we go. Now this is not meant to be confusing. Okay? This is not meant to be confusing. So what do we start off with? We start off with 2 acetyl-CoAs, and then we add a third. So you put 2 acetyl-CoAs together and you get acetoacetyl-CoA. Then you add a third and you make this molecule 3-hydroxymethylglutarol-CoA. And then we go down here and we add some electrons and we get mevalonate. So all we're showing here is, so far we've used 3 acetyl-CoAs. There's 2 and there's the third, to make a molecule called mevalonate. And all this brown box in here is to show you where the carbons are. Okay? So all this activity, we've only harvested really 2 carbons. Okay? And so at the end of all this biosynthetic pathway, we're going to make an intermediate that contains 5 carbons. So all this work has been done for 5 carbons. And just remember, we need 27 carbons to make cholesterol. So that names are not important. You're never going to have to be asked to do this, to reconstitute this. I just want to show you that a steroidogenic cell can start off with acetyl-CoA. Okay? And these 2 carbons here, these 2 carbons here, these 2 carbons here, and these 2 carbons here come from one of those acetyl-CoAs. That's all it's showing. So all we're doing here is we've made mevalonate, so we're back up here with mevalonate. That's the same one there. Then we phosphorylate at the 5 carbons. So there's 1, 2, 3, 4. Let's see. Well, there's 1, 2, 3, 4, 5th carbon there. I guess they don't count that guy. Okay, so that's mevalonate 5-phosphate, mevalonate 5-pyrophosphate. We're back over here, mevalonate 5-pyrophosphate to make this, and then we take off a phosphate and a CO2. Okay, there we go. 1, 2, 3, 4, 5 carbons. Okay, So we made a 5-carbon molecule. We started with 3 acetyl-CoAs, and now we're here to isopentenyl pyrophosphate. Okay, so we've got 1, 2, 3, 4, 5 carbons of the 27 we need. Okay? We're not even close, are we? We're not even close, are we? So hang on. So notice this asterisk here is the 5 carbons we've got so far. Okay? So now we're over here in the double asterisks, and here's the 5 carbons we've made so far. We used 3 acetyl-CoAs. We went to mevalonate. We went to 3-phospho-5-pyrophosphate mevalonate, and then we made isopentenyl pyrophosphate. And then we go over here, we go over here, we go over here. Boom, bottom line. Here is, now we've got a bigger molecule, okay? And to make this molecule right here, who's starting to look like a steroid, which its name is squalene, and lo and behold, it's got 30 carbons, okay? Now remember we had 5 carbons here. That's why I have a 5C here. We got 5 carbons here. My point is to go from here to here to here to here. After we're all said and done, it's going to take 6 isopentenyl pyrophosphates to make squalene. And squalene has 30 carbons, and all we have to do now is get rid of 3. So we cut 1 off, we cut 2 more off, and lo and behold, who do we have here? Lord have mercy, who is this? This is cholesterol. Okay? Let's see if we can number the carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. I messed up, didn't I? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. No, that's 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27. Okay? So now we have something we can make steroids with. Okay? Now we have something we can make steroids with. Okay? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. There, that's better. Okay, so these numbers are important to a biochemist, not to necessarily to a reproductive physiologist, but to a biochemist. So, at the end of the day, a cell that makes testosterone in the testes and a cell that makes estrogen and progesterone or progesterone in the ovary can start off with what, folks? 3 acetyl-CoAs, blah, 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 all the way over here, and we've needed 6 of these intermediate 5 carbons, so 6 times 5 is 30. There's 30, take away 3, and what do we have? We have cholesterol. So if I were ever to ask you if Dr. Stanko's on a low cholesterol diet because of his heart condition, if I ever ask you that, are you worried about him making testosterone? And your answer would be no. Why? Because my Leydig cells, because I'm on a low cholesterol diet, that's not true though, I eat a lot of meat, but if I were on a low cholesterol diet, I didn't eat meat and I didn't eat eggs and I didn't eat anything with cholesterol in it, would you be worried? So if you're a vegan, would you be worried if you're not going to make enough estrogen, progesterone, and testosterone? The answer would be what? No. How come? Because the cells in the gonads can make cholesterol, honestly, starting with 3 acetyl-CoAs, which is 6 carbons, and turned into 5. We just got to do that 6 more times. So holy moly. So we really need about 18 or so acetyl-CoAs, so we can make 6 isopentenyl pyrophosphates to make squalene, and from squalene we all gotta do is just get rid of 3 carbons. Okay, is that wild or what? Okay, again, do not memorize it, just say wow. Steroidogenic cells can make their own cholesterol, but they would prefer to get it from the blood from the diet of the host animal. Alright, so once again, what are the 2 sources of cholesterol needed for steroidogenic biosynthesis, steroid biosynthesis? Well, we can either make it from acetyl-CoA, or we can get it from our diet, and it's carried in our blood, also stored in the liver. So what we don't take up can be stored in the liver also, and the liver can release it. But, you know, if you can get it for free from the diet, it's a lot less energy expended than if you had to make it yourself. Okay, if I'm a steroidogenic cell. So I would prefer to get it from the blood. Okay? Alright, let's go look at the actual first figure in your textbook. So once again, this is kind of what I showed you before, just a different way to look at it. So here we have a low density lipoprotein, and here's the receptor. Here's the protein receptor, just like a hormone receptor in the cell membrane. Okay, so what happens first? Here's number 1. LDL binds to the receptor on the plasma membrane and are internalized through not endocytosis in clathrin-coated vesicles. So here's a clathrin-coated vesicle. Okay, the LDL receptor complex dissociate inside the endosome. So we've got an endosome in here, which is a bag of enzymes. Okay, so the LDL receptor comes in, it falls apart in the endosome, and cholesterol is released by the cholesterol ester hydrolase activity, or CEH. Okay, so here comes the endosome. It's now a lysosome. What came out was a receptor. It got recycled. Okay, so there's a question mark here because HDLs don't carry much cholesterol, so we don't know really if a cell can get cholesterol from HDL. I guess if you have low LDL levels, I guess it could get it from HDL, but that's just a question mark. So what got recycled was a receptor. Okay, so intracellularly, number 3, cholesterol is either esterified with fatty acids by the enzyme acetylcholesterol acetyltransferase, or ACAT, and stored in lipid droplets. So really, cholesterol is stuck to a fatty acid and stored as a fat droplet inside a membrane inside the steroidogenic cell, waiting to be released again. Okay. Number 4, what's going on here? Okay, if it's not stored, if it's not re-esterified and stored, it's going to be transported, because that's an or. So 3 is an either or. So if this cell is actively producing steroids, then that cholesterol has to be transported to the inner mitochondrial membrane. Okay, and that's where a protein called STAR, steroidogenic acute regulatory protein. Remember, that protein was discovered by Dr. Stocko at Texas Tech. Okay, so right where my red dot is, that's where STAR works. And what STAR does, STAR collapses the outer mitochondrial membrane, which is here, and the inner mitochondrial membrane, collapses the two and allows cholesterol to get access to that first enzyme that we're going to need to start to convert it to a steroid. So that definitely, STAR, the enzyme where my highlighter is, STAR stands for steroidogenic acute regulatory protein. STAR is definitely controlled by LH, and it is one of the initial rate limiting steps in making steroids. Okay, we've got to get cholesterol to the mitochondria to cleave off that side chain, to cleave off those 6 carbons, so we can have pregnenolone. Okay, and another point here is, guess who's making their own acetyl-CoA in the endoplasmic reticulum, in the smooth ER? Guess who's making it? The steroidogenic cell. So the steroidogenic cell can make acetyl-CoA, which we need 3 to make that intermediate, and then we need 6 of those 5 carbon intermediates to make squalene, and from squalene we get rid of 3 carbons and we make who? Cholesterol. So there's a lot in this picture. There's a lot in this picture. But again, 1, 2, 3, and 4 are all proteins. They're all enzymes. Okay? And guess who controls enzymes? The hormones, LH and FSH. Okay? All right, so the first thing that has to happen, and we'll end this first part of the lecture, this part 1, with this. This is directly out of your text. Say, the first thing we have to do to cholesterol in the mitochondria is whack off that side chain. So it's called the side chain cleavage. So side chain cleavage is performed by an enzyme complex consisting of a specific cytochrome P450, which is a type of enzyme. So it's called cytochrome P450 side chain cleavage enzyme. So anytime you see P450 SCC, that stands for side chain cleavage. So that's an enzyme complex that's located on the inner mitochondrial membrane. Okay. P450s are a monooxygenase enzyme responsible for the hydroxylation of lipophilic substances like cholesterol. So in this example, RH is cholesterol, is a lipophilic substance. So to get cholesterol, to cleave off, or to convert it to pregnenolone, we need to cleave off 6 carbons and put an OH group in the place of that 22nd carbon. So since P450s are monooxygenases, guess what they need? They need a molecule of oxygen. They need some electrons, which are going to be brought to the reaction by NADPH. Need some hydrogen ions. And so this would be cholesterol. This would be pregnenolone. That would be pregnenolone. Okay? That would be pregnenolone. Okay? So, you know, I know it's kind of funny to look at that representation, but basically it added an oxygen. Okay, cleaved off the side chain and added an oxygen. So, if you will look on the first page of your handout, you will see that carbon 20 of pregnenolone, carbon 20 of pregnenolone contains an oxygen. Okay? And that's where that oxygen was added. That's where that oxygen was added. It's not an OH group like shown here. It's a double bond oxygen. And so that's kind of what happened. We cleaved off the side chain and we added an oxygen and a hydrogen ion and an electron. Okay? I'm not making this biochemistry class, but I want you to know it's quite an ordeal to knock off that side chain. All right. So what do we need? We need 2 electrons for each hydroxylation. And where do we get those 2 electrons? We get them from NADPH, which is nicotinamide dinucleate phosphate. You should have heard this before in other classes, NADPH. In the case of mitochondria, P450 side chain cleavage enzymes, we're going to need some electron transfer carrier, 2 of them. One of them is called flavoprotein, which is a ferredoxin reductase, and FAD. So we need a flavoprotein called FAD. We need an iron sulfur protein called ferredoxin. And so we're going to take a look at where these electron carriers are located in the mitochondrial membrane. And if you want to look ahead, that's going to be figure 3 and figure 4. So basically, I'm just trying to show you, the chapter's trying to show you how we're getting this cholesterol molecule converted. Okay? So what I'm going to do is, on the second part of the lecture, I'm going to start with figure 2. Okay, I'll start with figure 2 on the second part of the lecture. All right.